Now we can discuss the second subtopic of muscle physiology, which is nothing but the molecular basis of muscle contraction. അപ്പോൾ ഒരു മസിൽ കൺട്രാക്ട് ചെയ്യുന്ന സമയത്ത് അതിൻ്റെ മോളിക്കുലർ ലെവലിൽ എന്തൊക്കെ സംഭവിക്കുന്നതാണ് നമ്മൾ ഈ വീഡിയോയിൽ ഡിസ്കസ് ചെയ്യുന്നത്. ആസ് യു നോ, ഇറ്റ്സ് വൺ ഓഫ് ദ ഇമ്പോർട്ടൻറ്റ് ടോപ്പിക് ഇൻ ഫസ്റ്റ് ഇയർ MBBS ഫിസിയോളജി, ആൻഡ് most of the time there will be a question from it. So coming to the topic, the molecular basis of skeletal muscle contraction is explained by sliding filament theory, which is also known as Ratchet theory or walk-along theory. And മൂന്ന് പേരുകളുണ്ട്, and it was proposed by the famous Huxley and Hansen. So as I said, there is an edge zone which is a relatively lighter zone in the center of an air band. അപ്പോൾ അതിൽ edge zone was coined by the famous scientist Hansen, who explained the sliding filament theory. By saying that, we can come to the topic proper. So there are 5 steps in a sliding filament theory. First one is the initiation of cross-bridge cycling, and second step is the formation of actin myosin complex. Third step is the power stroke, generation of the power stroke. Fourth one is detachment of myosin head and the cross bridge, and the fifth and final step is again reactivation of myosin head. Now we can come into the details of the first step, that is nothing but initiation of cross bridge cycling. So during initiation there are 2 main things happening. First one is, due to the action potential, there is a release of calcium ions into the sarcoplasm. So these calcium ions bind to the troponin C, because troponin C has high affinity to the calcium ions, and due to its binding there is a conformational change bringing about, and due to these changes, as I explained in the previous video, there occurs a exposure of active site on the actin. So active sites are now exposed. Now what happens to the myosin head? There is an intrinsic ATPase present on the myosin head. So due to the intrinsic ATPase, the ATP attached to the myosin head is cleaved into ADP and phosphate. So നമ്മൾ ആദ്യം തന്നെ നേരത്തെ കഴിഞ്ഞ വീഡിയോയിൽ പറഞ്ഞിട്ടുണ്ടായിരുന്നു, myosin head has 2 attachment sites. One site is for attachment to the ATP, and one side is to the attachment for active site on the actin. So നമ്മൾ ATP attachment സൈറ്റിൽ എപ്പോഴും ATP ഒന്ന് അറ്റാച്ച് ചെയ്യും. So due to the intrinsic ATPase activity of the myosin head, it is cleaved into ADP and phosphate, and these ADP and phosphate are just cleaved and they are stored in the myosin head itself. They are not yet released. So due to this intrinsic ATPase, it cleaves ADP into ADP and phosphate, and now the myosin head comes to its active state or charged state, which is also known as the cocked position of the myosin head. So once the ATP is cleaved, this myosin head there is a change in the position of this myosin head. After cleaving, it comes into an hyperextended position. So this myosin arm comes into an extended position, so that there is an angle of 90 degree. It was initially 45 degree. Now in a cocked state, the angle between arm and the tail is 90 degree. അപ്പോൾ there are 2 things. 2 things ആണ് നമ്മളിവിടെ, 2 മെയിൻ കാര്യങ്ങളാണ് ഇനീഷ്യേഷൻ ഓഫ് ക്രോസ് ബ്രിഡ്ജ് സൈക്ലിങ്ങിൽ ഹാപ്പൻ ചെയ്യുന്നത്. ഫസ്റ്റ് വൺ ഈസ് റിലീസ് ഓഫ് കാൽഷ്യം അയോൺസ്, ആൻഡ് ഡ്യൂ ടു ദ കാൽഷ്യം അയോൺസ് ഇറ്റ് ബൈൻഡ് ടു ദ ട്രോപ്പോണിൻ സി. അപ്പോൾ ട്രോപ്പോണിൻ സി ആയിട്ട് ബൈൻഡ് ചെയ്യുമ്പോൾ അവിടെ ഒരു കൺഫോർമേഷണൽ ചേഞ്ച് ഉണ്ടാവും ട്രോപ്പോണിൻ മോളിക്യൂളിൽ. ട്രോപ്പോമയോസിൻ മോളിക്യൂളിൽ ഒരു കൺഫോർമേഷണൽ ചേഞ്ച് ഉണ്ടാവും, ആൻഡ് ഇറ്റ് എക്സ്പോസ് ദ ആക്റ്റീവ് സൈറ്റ് ഓൺ ആക്റ്റിൻ. അപ്പോൾ ആക്റ്റിൻ ഫിലമെൻറ്റിലുള്ള ആക്റ്റീവ് സൈറ്റ് എക്സ്പോസ് ആയി കിടക്കുകയാണ്. പിന്നെ നമ്മൾ ഇനി നോക്കേണ്ടത് മയോസിൻ ഹെഡിലേക്കാണ്. അപ്പോൾ മയോസിൻ head, as I already discussed, there is an ATP binding site on the myosin head. Due to the intrinsic ATPase, അപ്പോൾ അതിന് സ്വതസിദ്ധമായ ഒരു ATPase ഉണ്ട് മയോസിൻ ഹെഡിൽ. അപ്പോൾ ഈ ATPase എന്താ, ATPനെ ക്ലീവ് ചെയ്യുന്ന സാധനമാണ് ATPase. അപ്പോൾ ATPനെ ക്ലീവ് ചെയ്തിട്ട് അത് ADP ആൻഡ് ഫോസ്ഫേറ്റ് ആക്കും. പക്ഷേ ഈ ADPയും ഫോസ്ഫേറ്റും still they are attached to the myosin head. They are not yet released. Due to this cleaving, there is a release of energy. Of course, there will be a release of energy while cleaving ATP. And due to this release of energy, the myosin head, which was previously like this, comes into a cocked state. Cocked state is nothing but this arm. This is the arm of myosin. This is the tail of the myosin, and this is the head of the myosin. Initially it was 45 degree. Now it extends to have a 90 degree between its arm and the tail. So this state is called as coked state or active site of the myosin head, which is known as charged state of the myosin head. So these are the two major things happening in the initiation of cross-bridge cycling. Now the second step is formation of actin-myosin complex. In the previous step, as I already mentioned, the active site on the actin is exposed and the myosin head has come into a state of active state. It is also known as the coked state. Now the second step is nothing but the formation of actin-myosin complex. So this activated myosin head attaches to the active site of actin, and due to its attachment to the active site, there is a release of the ADP and phosphate which was formed in the first step. Now the third step is power stroke. Once the active myosin head attaches to the active site on the actin, it brings about a conformational change in the myosin head, due to which the myosin head tilts towards its arm. During its tilt, the myosin head pulls the actin filament toward the center of the myosin filament, and it brings about the power stroke. So what happens in the power stroke? This attached, as we can see, this myosin head is attached to the active site on the actin, and due to its attachment, there is a conformational change happening on the myosin head, due to which the myosin head tilts towards the arm of the myosin, and during this tilt, it pulls the actin filament. So it pulls the actin filament toward the center of the myosin filament. It pulls the myosin head. Pulls the actin toward the center of the myosin filament. So it brings about a work because there is displacement, and this is known as work stroke or power stroke. And after this tilt, myosin head comes into a low energy state. Initially it was in a high energy state or coked state, and once this tilt happens, myosin head becomes into a low energy state. So these are the things which is happening in the power stroke. പവർ സ്ട്രോക്ക് എന്താണ് ഒന്നുകൂടെ എക്സ്പ്ലെയിൻ ചെയ്യാം. നമ്മൾ നേരത്തെ സ്റ്റെപ്പിൽ മയോസിൻ ഹെഡ് അറ്റാച്ച് ചെയ്തിരുന്നു ആക്റ്റീവ് സൈറ്റ് ഓൺ ദ ആക്റ്റിൻ. മയോസിൻ ഹെഡ് ഹാസ് അറ്റാച്ച്ഡ് ടു ദ ആക്റ്റീവ് സൈറ്റ് ഓൺ ദ ആക്റ്റിൻ. നൗ ഡ്യൂ ടു ദിസ് അറ്റാച്ച്മെൻറ്റ്, ഈ അറ്റാച്ച്മെൻറ്റ് മൂലം അവിടെ ഒരു കൺഫോർമേഷണൽ ചേഞ്ച് കൊണ്ടുവരുന്നു. ആൻഡ് ഡ്യൂ ടു ദിസ് കൺഫോർമേഷണൽ ചേഞ്ച്, മയോസിൻ ഹെഡ് ടിൽസ് ടുവാർഡ്സ് ദ ആം ഓഫ് ദ മയോസിൻ. അപ്പോൾ മയോസിൻ ഹെഡ് ആമിൻ്റെ അത് ഭാഗത്ത് ഡയറക്ഷനിലേക്കൊന്ന് ടിൽറ്റ് ചെയ്യുന്നു. ഡ്യൂ ടു ദിസ് ടിൽറ്റ്, ഈ ടിൽറ്റ് മൂലം എന്ത് സംഭവിക്കുന്നു? ആക്റ്റിൻ ഫിലമെൻറ്റ് ഈസ് പുൾ ടുവാർഡ് ദ സെൻറ്റർ ഓഫ് ദ മയോസിൻ ചെയിൻ, ആൻഡ് it brings a work done which is known as the power stroke or work stroke. And after this tilt, myosin head become a low energy state. Now we can come into the fourth step of the sliding filament theory, that is nothing but detachment of the myosin head. We have already seen that there is a work has been done. Now we have to detach the myosin head. Now we can discuss how this myosin head is detached. As we have seen in the second step, the ADP has released, ADP and phosphate has released from the myosin head. So there is a free site for the attachment of a free molecule of ATP or new molecule of ATP. Once this new molecule of ATP attaches to the myosin head, its affinity towards actin decreases. So it has low affinity to the actin, so it detaches from the actin molecule. അപ്പോൾ എന്ത് സംഭവിക്കുന്നതെന്ന് വെച്ചാൽ, മയോസിൻ ഹെഡ് ഓൾറെഡി വർക്ക് ഡൺ ഒക്കെ കഴിഞ്ഞു. ഇനി അതിന് ചെയ്യേണ്ടത് എന്താ? ഡിറ്റാച്ച്മെൻ്റ് ആണ്. അപ്പം ആസ് ഐ ഓൾറെഡി ഡിസ്കസ്, മയോസിൻ ഹെഡിന് രണ്ട് അറ്റാച്ച്മെൻറ്റ് സൈറ്റ് ഉണ്ട്. ഒന്ന് ആക്റ്റിനോട് അറ്റാച്ച് ചെയ്യുന്ന സൈറ്റും ഒന്ന് ATPനോട് അറ്റാച്ച് ചെയ്യുന്ന സൈറ്റും. ഈ രണ്ടാമത്തെ സ്റ്റെപ്പിൽ തന്നെ ADPയും ഫോസ്ഫേറ്റും ഓൾറെഡി ഡിസ്കണക്റ്റ് ആയിരുന്നു. അപ്പം ഈ ATP ബൈൻഡിങ് സൈറ്റ് ഫ്രീ ആയിരുന്നു. അപ്പം ഈ ATP ബൈൻഡിങ് സൈറ്റിലേക്ക് ന്യൂ മോളിക്യൂൾ ഓഫ് ATP വരുമ്പോൾ ആക്റ്റിനോടുള്ള മയോസിൻ ഹെഡിന് ആക്റ്റിനോടുള്ള അഫിനിറ്റി കുറയും. അപ്പോൾ ATP myosin head molecule has low affinity towards the actin, and thus it detaches from the actin. Now we come to the final step, that is reactivation of the myosin head. Now this detached myosin head has intrinsic ATPase, and which it cleaves the ATP into ADP and phosphate. So wherever there is cleaving of ATP, there is release of energy, and once again this myosin head myosin head comes into the cocked position. So it's ready to begin a new cycle. So this is the things that is happening in the fifth step. So intrinsic ATPase, the detached myosin has intrinsic ATPase, which cleaves ATP into ADP and phosphate, and myosin head gets activated once again and goes into the cocked position. This is the thing. These all are the things happening during the sliding filament theory. Now we can look into this cycle once again. First of all, this is the actin filament, and it has an active site. This is the myosin molecule. It has a head, it has an arm, and it has a tail. This myosin head has intrinsic ATPase, which hydrolyze the ATP into ADP and phosphate, which are still attached to the myosin head. And during an action potential, these calcium ions are released, and these calcium ions are responsible for the exposure of active sites on the actin. So due to the release of ATP, due to the breakdown of ATP, this myosin head comes into an cocked position and attaches to the active sites on the actin. And due to these attachment, ADP and phosphate are released, but also it produces a conformational change in the myosin head, due to which it tilts towards its arm. So during this tilt, it pulls the actin towards its center. So there is displacement and there is force, which is equal to work done, and this work done is called as the power stroke. It is also known as the work stroke. So after this tilt, the myosin head comes into a low energy state, and also there is a new molecule of ATP being attached to the ATP binding site. Due to this attachment, there is detachment of the myosin head from the actin binding site. So now the myosin head has detached, and new ATP molecule has bind, and this detached myosin head has ATPase activity, and ATP is again broken down into the ADP and phosphate, and again it bring about a conformational change, so that it comes into an active state or hyperextended state or cocked position, and also it again bring about the new cycle. So let's discuss it once again. The free myosin head has intrinsic ATPase, which hydrolyzes the ATP into ADP and phosphate, and it comes into an active state, or it is also known as a cocked state of myosin head. And due to the release of calcium, which binds to the troponin, and there is exposure of the active site on the actin. Now this cocked myosin head attaches to the active site on the actin. Due to its attachment, the ADP and phosphate are released, and also there is a conformational change on the myosin head, due to which it bends towards its arm. Due to this bending, there is a pulling of actin towards the center, and after this is known as the power stroke or the work stroke. And after this pull, the myosin head comes into a low energy state. Once it has come into a low energy state, there is a new molecule of ATP which attaches to the myosin head. And as you know, this ATP myosin head compound has low affinity towards actin, thus there is detachment. Once again, this ATP is hydrolyzed and it forms an ADP and phosphate, and the myosin head goes into a cocked position. So these all are the steps which is happening during a sliding filament mechanism or sliding filament cycle. Now we can look into the mechanism of muscle relaxation. As I already said, calcium ions are very much important for muscle contraction. This calcium binds to the troponin tropomyosin molecule, and it helps in the exposure of active sites on actin. So calcium is responsible for making the active sites on actin to be exposed. So once the action potential is stopped, these calcium ions are pumped back into the sarcoplasmic reticulum, and calcium ion concentration in the sarcoplasm decreases. So there is increased sarcoplasmic calcium level during muscle contraction. Once the action potential stop, there is activation of calcium ATPase and the calcium is pumped back into the sarcoplasmic reticulum. So decreased sarcoplasmic calcium level. So there is detachment of calcium from troponin and there is cessation of actin-myosin interaction, and it helps in the muscle relaxation. So calcium was responsible for binding to the troponin and exposure of active sites on the actin. Once the calcium level is decreased, calcium level കുറയുമ്പോൾ എന്ത് സംഭവിക്കുന്നു, ആ troponin molecule again attaches on the active sites on the actin and there is no site for the myosin head to bite. It results in the cessation of actin-myosin interaction and there occurs the muscle relaxation. I think it is very clear to you. So whenever the calcium ions decreases, this troponin molecule once again goes back to its original position and it covers the active site on the actin molecule. So once it covers the active site, there is no site for attachment of myosin head and it brings about the cessation of actin-myosin interaction and it results in muscle relaxation. Now we can come to the applied aspects of the sliding filament theory. First of all, we can look to the rigor mortis. Rigor mortis is nothing but the stiffness of the muscles and joints which is occurring after the death. So after the death, our body goes into a state of stiffness of the muscles and it's known as the rigor mortis. So what is the mechanism behind this rigor mortis? It can be explained by using the sliding filament theory. Soon after the death, the cell membranes become highly permeable to the calcium. So whenever the calcium ions are high, it promotes the formation of actin-myosin complex and it results in the contraction of the muscle. Now this contraction, this rigor mortis is present only for few days after the death. So what is the mechanism behind this stoppage of rigor mortis? We can look into it. Now for muscle relaxation, calcium ions should be moved out of the muscle and it requires ATP because calcium ATP is the pump which pumps back the calcium ions into the sarcoplasmic reticulum. After that, there is no ATP remaining and no new ATP cannot be formed. So muscle remains in a contracted state. But due to the decomposition of the body, these lysosomal enzymes are released, and due to the lysosomal enzymes, there is hydrolyzing of the muscle proteins. Due to this hydrolysis, there is breaking down of actin-myosin complex. So lysosomal enzyme is responsible for the breaking down of actin-myosin complex and it relieves the stiffness of the muscle after few days after the death. Now what is the medico-legal importance? So this rigor mortis starts, as I already mentioned, it starts 10 minutes or 10 minutes to 3 hours after the death and it depends upon the environmental temperature. And it peaks at 12 to 24 hours and it can continue for 1 to 3 days. So by using this, the rigor mortis, we can determine the time since death. So the medico-legal importance of rigor mortis is that it helps to understand the time since death. I think everybody is clear regarding this rigor mortis and its medico-legal importance. There are two more applied aspects for sliding filament theory. It's none other than the micromotor unit formed in the polio and the muscular dystrophy, which occurs due to dystrophin gene mutation which connects the actin to the muscle cell membrane. There are two types of muscular dystrophy. One is Duchenne muscular dystrophy and another one is Becker's muscular dystrophy, both of which occurs due to the dystrophin gene mutation. And Duchenne muscular dystrophy is more severe than the Becker's muscular dystrophy, and both are X-linked recessive and are seen in male child only. So you will learn this in the coming years. I have included this for your understandings. So I think sliding filament theory is very well understood for you, and its applied aspect, especially the Ragar mode, is very well understood for you, and its medico-legal importance. And there are two more applied aspects, which is macromotor unit formed in polio and the muscular dystrophy, which occurs due to the dystrophin gene mutation. And that's all about this topic. And I hope you will do very well in this coming physiology exam.